Okay, yeah, I'm recording. Back to
Okay,
you.
good. All right, we're on. Good, thank you. Good. All right, this is. Okay. Okay. Sorry about this. Let me just get to that. Okay, so what I'm basically doing right now is covering the last of the two major spinal projection systems that I want to cover. If you remember in the last class, we went over the dorsal column / dorsal column postsynaptic system. And then we went over the anterior spinal thalamic tract. And then we went over the lateral spinal thalamic tract. So now we're going to go through the anterior and posterior spinocerebellar tracts. So when we were looking at spinal cord organization, recognized that the five most superficial laminae 1 through 5, were rather very sort of clear in terms of using that word called lamina, which means layer and layers of cells. And what we saw was that in layer 1, it was the posterior marginal zone. Layer 2, you had the substantia gelatinosa. Lamina 3 and 4, you had the proper sensory nuclei. Then lamina 5, you had the medial and lateral reticular nuclei. And of course, a lot of that would cover spino-spinal communication with layer 1 through the zone of Lissauer. And then what we have basically seen is that layers 2 and 5 are the major areas by which their axons come out and form the anterior spinothalamic tract, the lateral spinothalamic tract. Then laminas 3 and 4, the axons of those neurons largely form the anterior spinothalamic tract. Obviously, 1 through 5 also contribute into the dorsal column postsynaptic system. So now we get to layer 6. Layer 6 is not so much as a layer as an area. And we talk about in layer 6, we call it the base of the dorsal horn. So you can see the name of the sort of a general area. But a major nucleus, which is found in only a certain number of segments in the spinal cord in layer 6, is the nucleus of Clark. And the nucleus of Clark can be found around L1, L2, and it can be found as high as C8. So what you can see is that the upper part of the lumbar cord and the whole thoracic cord, and then the very caudal part of the cervical cord has this nucleus. Now, when you basically look at lamina 6, what you can probably tell is you're still getting some fibers coming in from the dorsal root ganglion, okay? But in lamina 6, what you're also getting are fibers that are largely entering in through what we call that emerging lateral horn, which is structurally found mostly in the thoracic cord. And what is the difference between fibers coming in through the dorsal horn or dorsal root ganglion? They are primarily somatosensory in all the different combinations and permutations that I covered in the last class. But now in the thoracic cord, especially the lateral horn, what we have are largely what we call viscerosensory fibers. And viscerosensory fibers are either coming a lot from smooth muscle, or they are coming from deep areas within the body that provide sensory input, and they're providing sensory input about the positioning of the body, or shifts in the positioning. So therefore, these viscerosensory fibers and the few somatosensory fibers that come in and synapse in the nucleus of Clark in lamina 6 are giving information that really has a lot more to do with what we call proprioception or a proprioceptive kinesthetic feedback. Again, proprioception is telling you about where your body is in space rather than some sensory external event. Okay? And the second thing is that that this dorsal horn, this nucleus of Clark is getting viscerosensory input. So again, you're getting not so much proprioceptive, but also interoceptive types of information. So now this information is now coming into the nucleus of Clark. And the nucleus of Clark, these large cells, are going to form the major integrative input going into the cerebellum from the lumbar cord, a little bit from the sacral cord, and then very much from the thoracic cord, not the cervical cord. So the interesting thing is that these spinocerebellar fibers, these fibers that are the axons of the nucleus of Clark, are fairly myelinated fibers. They can be A delta, they can even be A beta. So that information is going up quickly. But what it does is it shoots it up along two parallel pathways. One called the posterior spinocerebellar tract, and the second, which is the anterior spinocerebellar tract. And very often what we can see is that these two parallel pathways are starting roughly in the same part of the spinal cord, some thoracic segment, and they are ending up in the cerebellum. Now, the somatotopic organization that we have basically seen with the dorsal column system is not as sharp here, but there is a sort of a segmentation of that. It's not been as studied as the dorsal column system. So why would we have two pathways that are literally identical in their conduction velocity? They're literally identical in where they go up to the cerebellum. It's that they actually take a shorter path and a longer path. So the spine, the posterior spinocerebellar tract is going to originate from fibers in the nucleus of cells in the nucleus of Clark. And what these fibers do is they go out ipsilaterally out onto the sort of dorsolateral surface of the spinal cord from where they originated. So they then form this posterior spinocerebellar tract. And from there, that posterior spinocerebellar tract traverses the spinal cord. And then when it comes up through the medulla, we are now, that posterior spinocerebellar tract is now in what we call the inferior cerebellar peduncle. So here is a situation where we can really use the verb consists of or comprises. So what I'm basically telling you right now is that the inferior cerebellar peduncle is comprised in part by the posterior spinocerebellar tract, because you're just going straight through, going into the medulla of the inferior cerebellar peduncle, and then in the caudal pons, what happens is that the inferior cerebellar peduncle enters into the vermis of the cerebellum. And what is the vermis of the cerebellum? Deep cerebellar nuclei. And when we go through the deep cerebellar nuclei later in the course, you'll see what their importance is in terms of input and then output that's coming from the cerebellum proper. So, in addition to the nuclei of Clark forming this ipsilateral posterior spinocerebellar pathway, it also gives rise to a second pathway that pretty much at the level that the nucleus of Clark is found, the fibers now go across the anterior white commissure and then form the anterior spinocerebellar tract. Now look at the anterior spinocerebellar tract. It ascends through the spinal cord. It ascends through the medulla. It ascends through the pons. And then it finally comes up to and enters the superior cerebellar peduncle. So the superior cerebellar peduncle is comprised in part of the anterior spinocerebellar tract. Now, what does the superior cerebellar peduncle do? It then decussates and then enters into the cerebellum much more anteriorly. Okay? So, what we have are two distinct spinocerebellar and anterior spinocerebellar tracts emanating from the nucleus of Clark. The posterior spinocerebellar tract has a shorter trajectory, entering the cerebellum and the vermis basically through the inferior cerebellar peduncle. The anterior spinocerebellar tract crosses the anterior white commissure, then travels upwards into the superior cerebellar peduncle, where it decussates a second time, and then enters the cerebellum. So both of the tracts, if you're following this, both spinocerebellar tracts innervate the ipsilateral cerebellum. That is important to note, that both spinocerebellar tracts innervate ipsilateral cerebellum, with the posterior spinocerebellar tract always being ipsilateral to its origin, and the anterior spinocerebellar tract doing a double decussation, crossing to the opposite side, coming up, then crossing back again, and then plunging down into this vermis of the cerebellum. So one of the questions is, why would you have two of these systems? One argument might be, well, one system is older than another. But the answer, that's not a good answer, simply because it appears on a phylogenetic way that both of these tracts appeared pretty much at the same time. So is there some type of functional significance to that? And what would be the nature of the functional significance? And if you basically look at it and you basically argue that a signal is coming out, two signals are coming simultaneously out of the nucleus of Clark, but they take two different pathways. The length of time it takes those two signals to come out of the common place and enter into the cerebellum are different, simply because the posterior spinocerebellar tract, by always being ipsilateral and going in a direct pathway through the inferior cerebellar peduncle into the cerebellum, happens quicker than the longer, more laborious pathway of the anterior spinocerebellar tract that has to cross the anterior white commissure, then go up contralaterally, go all the way through the medulla and the pons into the caudal midbrain, and then where it enters the superior cerebellar peduncle, decussates a second time, and then comes back down into the cerebellum. So there is some argument here that these two signals that emanate from the same place but arrive in the cerebellum at two temporally different things may be some mechanism by which the cerebellum, quote unquote, tells time or establishes a relationship of either the somatosensory input that it's giving or the viscerosensory input that it's given. And what type of input is that? It's either interoceptive input or proprioceptive input. So there's again a couple of things that I want to state with you. Obviously, there's a whole bunch of other pathways when I go through this course that I don't cover, simply because there's just a certain amount of things. The course, after all, is called basic neuroscience. It's not all of neuroscience. So what you're basically learning here is that the inferior cerebellar peduncle is comprised in part of the posterior spinocerebellar tract. The superior cerebellar peduncle is comprised in part of the anterior spinocerebellar tract. One other fiber area that I wish again to emphasize is the anterior white commissure. The anterior white commissure, now that we've covered a number of these ascending tracts, you will realize contains three different pathways: the anterior spinothalamic tract, the lateral spinothalamic tract, and the anterior spinocerebellar tract. All of them traverse the anterior white commissure. So of course, if we now start to put a couple of things together, so like if we basically look down here in the spinal cord and we look at where the anterior white commissure is, what is it basically close to? It's basically close to the anterior median septum, okay? And what is one thing that we know that is in the anterior median septum in the thoracic cord is we know that the anterior radicular artery is there. So if there was some insult to the anterior radicular artery and it affected the anterior white commissure, what could we basically expect? What we could expect is some type of damage or interruption to three of the major tracts that cross near the level at which, let's say, that insult happens. So that's the lateral spinothalamic tract, the anterior spinothalamic tract, and the anterior spinocerebellar tract. Then what you can basically look at that particular level of the spinal cord, if there was damage there, what type of functional significance would this have for all different types of sensory information? Well, by affecting the anterior spinothalamic tract, we would be affecting gross touch, gross temperature, pain, and whatever, gross touch and gross pressure. And then with the lateral spinothalamic tract, we'd be affecting pain, and you'd be affecting it basically because that pathway has members crossing both ways. You would have effects on both of the sides. Plus, you would have this disruption of the anterior spinocerebellar tract, where the posterior spinocerebellar tract is working because it's not affected, but the anterior spinocerebellar tract is affected. So what you may end up with is some problem with timing or coordination. So you can basically look very often as you go through this course, you can basically look at all of these pathways and basically ask yourself a simple question. What would happen if there was damage or destruction here? What would this do? What would that do? What would the other thing do? And basically through detective work, you could basically start to investigate the deficits. Then, of course, you might be dealing in a thing where you're, let's say, working in a neurology service or a psychiatry service, and all of a sudden you basically start to see a person exhibiting symptoms. And by exhibiting the symptoms, if there are some area like the somatosensory or viscerosensory or proprioceptive or these types of deficits, you then can go back and work backwards and say, based on these symptoms, we would think that there is something wrong here, and then investigate it. So it goes in both directions. You're basically going from an actual visualization of damage to a symptomatology, or a symptomatology back to where the particular part of the brain is disrupted. So there we have the anterior and posterior spinocerebellar tracts. So again, I'm not going to cover these here, but or you're not going to be responsible for it on the exam, but I want you to know that they're there. Is again, we have spino-reticular and reticulospinal tracts. And of course, here is a perfect example of the importance of our friend the hyphen, because in spino-reticular, we see that that's an ascending afferent type of symptom, a system in which there is primarily somatosensory information being carried from a level of the spinal cord up into the medullary, pontine, and midbrain reticular formation, and we'll talk a lot more about the reticular formation today. And then the reticulospinal, so the origin of these fibers is in a reticular formation, and it is a descending tract down into the spinal cord that is probably playing a very major role in motor. And if you, so now, when I sort of introduced the efferent spinal projection systems, I basically pointed out to you that there were 3 of them: a reticulospinal tract that is playing a role with the proximal limbs around the shoulder and upper arm. So it's going to be very important in things like reaching. Then we had another system called the rubrospinal tract, which is the red nucleus that has direct connections from the basal ganglia. And of course, this is distal movement and finer movement using the fingers. And then, of course, we then have the corticospinal tract, which is going to be modulating and integrating both the reticulospinal proximal movements and the rubrospinal distal movements. And how did they do that? Not only does the corticospinal tract come down, either through the lateral corticospinal tract and anterior corticospinal tract, but that there is also a corticobulbar system that will literally modulate the reticular formation. And what you're going to basically learn is how a lot of parts of the motor cortex and other parts of the cortex then modulate a major structure called the basal ganglia, which is made up of the caudate and the putamen and the globus pallidus, and where the cortex is basically modulating the activity of the caudate and putamen. The caudate and putamen is modulating the activity of the globus pallidus, and then the globus pallidus is sending major outputs to the red nucleus. So you have an indirect type of modulation of cortical output, modulating these distal movements. So now let's look at this. And here what we have is, again, on the sensory side, you can see these interoceptive and proprioceptive mechanisms of muscle spindle fibers, which are excuse me, which are sensory. And what you can basically see here is that a primary muscle spindle controls the muscle length and speed, whereas the secondary controls stretch and muscle tension. So here you're getting a sense of how fast the muscle is changing, and over here you're getting how much the muscle is changing. So all muscle perceptions basically come from these spindle fibers, okay? And here you can basically look at a whole bunch of the underlying physiology of this. So you basically see with the speed is measured by amplitude, and you can see the receptive potential, whereas stretch is a much longer lasting type of effect, but the changes that happen are much smaller. Okay, then what we have is, again, in proprioception, is basically looking at an interaction between deep tissue, which basically is made up of tendons, and the interaction between these tendons and skeletal muscles. And what you have is the Golgi tendon organ that basically measures Golgi force and contraction. So again, what you're basically looking at here, and remember, look at that name, Emilio Golgi. I don't know how many of you were able to attend. I think a number of you attended Josh Brumberg's talk in the Behavioral Neuroscience seminar series. By the way, if there are those of you who don't know about this and want to know something about it, please tell me, and I'll make sure that the person who runs this, Desiree Bird, will include you. But with Josh Brumberg, when he did a beautiful history before his talk, he basically talked about two of the giants of the field. And one was Emilio Golgi, and the other was his student, who then sort of became his rival, Ramon y Cajal. And what you basically had was an argument about so-called neuron doctrine and the whole aspects of this. And Golgi obviously had created the stain by which you could start to study individual neurons, but he also did many things in terms of sensory information. So he identified not so much the somatosensory receptors, which were much easier and much more attainable. He sort of went in and looked in the deep muscle in terms of how these things moved in the Golgi tendon organ. So again, we have that major form of interoceptive and proprioceptive information. So now I go into the corticospinal tract. And the point of the matter is, I've been talking about the corticospinal tract ever since we started the class. Roughly an hour into the first lecture, I basically showed you a diagrammatic demonstration of the superior surface of the brain. And what we basically looked at were two incredible landmarks: the interhemispheric fissure that divided the two hemispheres, and then we saw this other deep sulcus come off on the superior surface and go down the lateral surface called the central sulcus. And of course, when we looked at the central sulcus, we saw two adjacent gyri, the precentral gyrus and the postcentral gyrus. And of course, when we looked at the precentral gyrus, the first thing I told you about was its alternate name, which of course is primary motor cortex. And from there, we started to understand the power of these cortical structures in terms of modulating movement. So the corticospinal tract that you can see, that is in the precentral gyrus, that is on the superior and then lateral surface of the brain, all the way down to the central sulcus, what we have is an enormous number of cells right here, and that these cortical layers, there's 6 cortical layers, and what we're going to basically find is that 2 of these layers in layer 3 and layer 5 are going to be primarily made up of cells. And these cells are very distinctive. They're large cells, and they have a sort of unique shape. What they are what are called pyramidal cells. So they basically look very often like upside down pyramids. And although there are some pyramidal cells that are not inverted, and Brumberg talked about those. But if we look at the predominant cell type, which are these pyramidal cells that are found in layer 3 and 5, what they basically have is a very interesting structure. They have a very large and protuberant apical dendrite, a dendrite that literally looks like a tree and goes up. So if the cells are found in layers 3 and 5 of the precentral gyrus, their dendrites move up into layers 2 and 4, okay? And then these dendrites, not only that apical dendrite, it not only has just that tree, but it then has branches, and then from the branches, it then will have little offshoots. And what you have is an enormous dendritic branching. And these are the inputs. They can get inputs from a whole group of cells that are traveling cortico-cortical, going across and synapsing on these apical dendrites. And of course, if you look at that pyramidal cell, and you basically look right near the base of that apical dendrite, you will find a whole bunch of receptors. And then you will find receptors all the way up through the apical dendrite. And of course, this becomes the poster child of the importance of the location and type of a receptor that can be on a neuron. And the point is that when we basically look at that neuron and ask whether it fires, we find that it is a result of some type of series of algebraic summations. And what are the algebraic summations? One is a spatial summation. So we can have a basic rule that where is the decision for a cell to fire going to happen? It's going to happen in the area, in the base of that pyramidal cell, before it forms its axon, called the axon hillock. So if there are receptors in very close apposition to the axon hillock, that receptor, whether it is excitatory or it is inhibitory, is going to have far greater influence in whether that cell fires or not fires than if the receptor is found way out on a dendritic branch of the apical dendrites. Okay, so there is the process in spatial summation of where are those receptors located and whether they are excitatory or inhibitory. Then you have, over a very brief period of time, in the areas of milliseconds or even less, you then have a process called temporal summation, in which that receptor can be occupied, thing taken in, and then another receptor and another receptor, and that is going, whether those receptors are inhibitory or excitatory, are going to make that type of decision. So you have this very elaborate type of synaptology in lamina 3 and lamina 5 of the precentral gyrus. And then, so therefore, particular cells may fire and other cells may be inhibited. So therefore, what you now start to get is a specificity. And that specificity, again, is what? Somatotopically driven. It is somatotopically driven simply because on the superior surface of the brain, we have the innervation of the lower appendages, then the trunk, and then on the lateral surface, the upper appendages and the head. So now all of these fibers will now come and they will join together from the postcentral gyrus and form the anterior limb of the internal capsule. So the a vast majority of the anterior limb of the internal capsule is what? Corticospinal fibers. So what are those fibers doing? They are moving from the superior and lateral outer surface of the brain, moving inward. So they are either moving inferiorly or moving medially through this anterior limb of the internal capsule. And then eventually you get to the inferior surface of the brain, in which now these fibers will now turn and move caudally. And that turn has a name. That turn is called the crus, C-R-U-S, cerebri, Cerebri. So it's a literal turn of the fibers moving inferiorly to now the fibers moving caudally. So now the fibers now form, they now enter the midbrain. It's the same fibers, and they are in the cerebral peduncle, which is in the midbrain. So they form the inferior surface of the midbrain, called the cerebral peduncle. And of course, what is the nature of these fibers? The nature of these fibers is, again, they maintain their topographical organization, okay? So now, as we run through, the midbrain, we now enter the pons. Do we see the corticospinal tract on the inferior surface of the pons? No, because a second fiber tract, the middle cerebellar peduncle or brachium pontus, comes in and is moving medial laterally. So in the pons, we call the corticospinal tract the corticospinal tract. Then when we get into the medulla, again, the corticospinal tract is now observed on the inferior surface of the brain, just like we saw in our first couple of lectures. And now we are calling this a separate structure called the pyramids. And here is the pyramids of the pyramidal tract. It's all the same tract, different names. But what happens in the caudal medulla? What happens in the caudal medulla, in keeping with our understanding of motor function, such that the left side of the brain is controlling the right side of the body, and the right side of the brain is controlling the left side of the body, 95% of these pyramidal fibers will decussate, and then in the caudal medulla and upper cervical cord, and then they will decussate and move into what we call the lateral corticospinal tract. 5% of the fibers will continue in their caudal trajectory in the same place, and we call that the anterior corticospinal tract. And of course, the anterior corticospinal tract will then innervate alpha and gamma motor neurons, and so will the lateral corticospinal tract at all of the spinal levels. So now, in addition to the corticospinal tract coming out of the precentral gyrus, the corticobulbar tract also comes out of the precentral gyrus, and they again go through the posterior limb, the anterior limb of the internal capsule. They then start to synapse all the way along. So one thing, look at this very interesting synapse. You have a synapse here that in which corticobulbar fibers, motor output, is synapsing around the medial lemniscus. What does that mean? It's modulating in some way medial lemniscal somatosensory input, because what's in the medial lemniscus? The dorsal column system and the anterior spinal thalamic tract. It also will modulate and synapse on the substantia nigra that we'll be talking about pretty soon. And then you can see it interacts with the midbrain tegmentum. It then interacts with the reticular formation in the pons, and then in the reticular formation in the medulla. And then it's going to interact with a very interesting structure that we'll be talking about very quickly, called the inferior olivary complex. So this corticobulbar output is motor output that is then modulating midbrain structures, pontine structures, and medullary structures, and can exert a quite direct role on reticulospinal output. So this is how these systems sort of work together. So that is the end of that particular lecture here. And that is going to be the major things. This is the last part of the stuff that can be covered in especially part 2 of exam number 1. So if we're now going to look at, and I think I'm looking at, we have 29 participants, and this is getting all recorded and whatever. I think at this point, this is a very good point at which to talk about part 2 of the exam. So in part 2 of the exam, it is going to be a written part of the exam. You're going to be submitting your answers to me via email and via a Word file, okay? Now, in a Word file, not only can you just type out the answers, make tables, do whatever the hell you want to go, but what you can also do is you can construct some type of a pictorial representation of the answer and then place that in. Now, the most important thing is that if you're going to use that option, and for some of the options like the projection pathways, or looking at the ventricular system, or maybe looking at certain aspects of the blood supply, it may be much more advantageous for you to do the classic picture is worth 1,000 words. But what you have to do is you just don't give me a picture. You give me a picture that's labeled, and then maybe put in explanations below the picture as to what that picture actually means. So, until I'm sorry, this is what happens in life. You have a person who you basically look at in terms of some type of career, and the person has an incredible career, and then shit happens. Or put it this way, you find out shit about the person. And the person I'm talking about right here is a gentleman by the name of Kevin Spacey. Kevin Spacey, up till about 2.5 years ago, was doing year after year of this incredible show called House of Cards, in which he manipulates his way to the presidency of the United States. And you watch this person. And of course, he was in many of the movies. But one movie that always struck out to me was a movie called The Usual Suspects. And the usual suspects were that this crime was basically happening. And what the cops do is go round up the usual guys we round up, and they bring them in together. And it's this incredible group of actors that come in, and headed by Kevin Spacey, who is plays a schlub named what was his name? Something Kent or something like that. He's like a nobody. But what he actually really is this unbelievable, violent, horrible, but genius type of person by the name of Kaiser Sose. And if you've never seen the movie, and even though it might be politically incorrect because you may never want to see anything that Kevin Spacey ever does, God Almighty, watch that movie. It's incredible. I always love that. So what I love is the usual suspects of that movie. And where did the name of that movie come from? It came from one of the most classic American movies ever made, with Humphrey Bogart and Ingrid Bergman and Kurt Honreid, called Casablanca. And in Casablanca is they're under the obvious overall control of the Nazis during World War II, but they're under the local control of what they call the Vichy French. And the Vichy French police force is headed by a guy named Claude Rains, who basically plays this inspector, a corrupt guy and whatever. And what he does every so often, just to placate the Nazis, is he goes, round up the usual suspects, and they bring people in as to whether or not the people did it or not. It was just a very intellectually lazy way of doing things. But I always loved that term because whenever I've been teaching over a 40-year period, very often with any kind of student, you basically have to go, they basically go, well, what's going to be on the exam? And the point is, you find an unbelievable range of people's ability to try to figure out what's going to be on an exam. So I'm going to basically say something. My favorite doctor, I think, as I've told you, is Dr. Seuss. And the reason Dr. Seuss is my favorite doctor is because he had a character. And that character's name was Horton the Elephant. And what was Horton the Elephant? He had a motto. He says what he means, and he means what he says. So when I teach you something, I can't teach you everything. So the things that I'm trying to teach you and get across are the things that I think are very, very important that you can carry away with you through your career. Yes, you'll find other things and whatever, but here I think are the important things. So in the keeping of saying what I mean and meaning what I say, when I give an exam, I'm not going to give you an exam on minutiae or exceptions to the rule. I'm going to give you an exam on what I think is important and what I think is important, the stuff that I stress. So what I want you to do, and I think I did this already, is figure out for yourself, what are the usual suspects? Well, if you basically looked at forgetting about identifications, which is going to happen on part one of the exam, in which I point at this and ask you this and ask you that for 40 questions. In part two, we're looking at these sort of larger concepts. So again, a very important thing that I went through were the cranial nerves. I went through encephalic development. I then went through the ventricular system and how that all ties in. I then went through the whole blood supply, particularly the arterial blood supply. Then we went through spinal cord organization within the gray matter, of course, Rex's laminae. And then you have this whole display of the dorsal column postsynaptic system, the lateral spinothalamic tract, the anterior spinothalamic tract, the anterior and posterior spinocerebellar tracts, and then the corticospinal and corticobulbar tracts. So what you can basically see is that there, for the remaining 60 points of the exam, you can see pretty easily what are the things that I'm going to ask. Now, under typical circumstances, you would come in on a day like this. If we were all in person, I would have gone through part one of the exam with all of you, the 40 questions, asking people to look up when they finished writing their answer. And then I would give you a certain amount of time to answer part two. Now you're going to be able to answer part two, and you're going to be able to answer it open book, okay? So the important thing here is I'm hoping that everybody is going to answer part two and know where to look. I'm telling you already, you can start to lay out your answers even as you have it right here. However, whereas before, if person X didn't mention this or that, maybe I'd give you a quarter point off or a half a point off or something like that. Here, given the fact that you have all this information in front of you and can put the things together, I want to make sure that you do a full answer. So if you do if you don't answer a particular part of the question, you're going to lose points from that. But that's the only way you're going to lose points. So the other thing, in order to do this and to keep the idea of when what we're going to do is I'm going to give everybody the exam all at the same time. And then I'm going to give you a 72-hour period to send the exam back to me. Now, some of you might send the exam back to me within an hour. God bless you, okay? You don't have to do that. But within 72 hours, you have to answer the question. So what I want you guys to do is designate someone who's going to collect the preferred times. Talk among yourselves as to when you want to see that exam. So that can happen as soon as today, October the 1st. Or you might decide, no, I don't want to see the exam until like October the 6th, okay, as a group. So if you see it on the 1st, what basically happens on the 1st, you would have to have it back by the 4th. If it was on the 6th, you'd have to have it back on the 9th. The last possible date that I want to see this is by Friday the 23rd. I would like everything handed in by then. But it's all of you getting all the material at the same time and then handing it back in at the same time. So one of you should write on behalf of everybody else, CC everybody else, and say, here's the time, and then I will provide you that exam, okay? So let me see, I see some chats down here. Let me see, you said I said 96 hours. Heaven forbid I have had a senior moment. 96 hours is perfectly fine. So 96 hours would be, if it was the last possible date, the 23rd, that's 24, 48, 72, you would get the exam on the 19th. On the other hand, one of the good things about my course is I give this quote midterm early simply because it's covering this gross brain and the spinal cord and the blood supply, the general types of things here. You have to also consider that a lot of you are full-time students and taking other exams. So probably somewhere in late October, you'll start to get a lot of midterms. So you may think that it's easier to give this exam a little earlier. I'm totally agnostic on that. But the latest I want to see it is the 23rd. Okay? And of course, by the 23rd, all of you will then be completely signed up for taking the taking part 1. So really by the end of October, you should really know where all of you stand with respect to part 1 and part 2. All right?
So just to clarify,
Go ahead. Sure.
Hi. Just to clarify, you want us to email you to
a Word file. It'll be very clear. When I send you the exam, there will be specific instructions. Yes. You would be sending me a Word file. Yeah.
No. I mean,
why a Word file? Because sometimes with PDFs, things get all screwy. You don't see.
No, I meant that you're going to send us the Word document let's say you were going to send it to us today, or do you want us to
No, I'm going
ask
to
you
send it.
to receive
I'm
it?
going to send it to you on the date that you want to receive it, and then 96 hours later, you're going to send me your responses. Yep.
Oh,
You don't
so we
get to
tell
see
you
the
when we
exam
want it, and
until
then 4 days
4
later we
days
give it.
before.
Okay.
I mean, unless I'm a horrible liar and you can castigate the hell out of me where I write surprise questions or something, I think I pretty much told you what's going to be on the exam. The specifics and extent of it will be found in the questions.
All right.
Pretty
I get it.
damn sure you're going to see a question on cranial nerves. You're going to see a question on encephalic development. You may see a question on ventricular system, on blood supply, on spinal organization, and on some projection system, okay? The
Okay,
specifics
all
of
right.
each of them, you will see 4 days before you have to hand it in.
All right, cool, cool,
Good.
cool.
Good. And it'll be your choice as to when you're going to see that. Okay, take a short break. I'll be back in about 5 minutes, and we'll start to jump into a totally new area that's not on the first exam called the medulla. Okay. I'll mute myself here. Okay, so I'm back. And what we're now basically going to be doing is entering the brain. And we are basically moving from the most caudal part of the brain going forward. And that, of course, is the medulla. So what we can basically see here is an inferior surface of the actually what we're looking at is, yes, an inferior surface of the brain. So we're basically seeing very major parts. Here is the deep cerebellum. And then what you're basically seeing right here is the pyramids. And then what you can basically see coming out are a variety of the cranial nerves that we're going to be talking about as we go through. So here is a schematic representation of the dorsal surface of the brain without the telencephalon. So what we basically have taken off is the cortex and the cerebellum. So what you're basically looking at is the dorsal diencephalon, which of course is the thalamus with the internal capsule. Then we're seeing the dorsal surface of the midbrain with the superior and inferior colliculi and a trochlear nerve and the superior cerebellar peduncle. Then we have the dorsal surface of the pons looking into the fourth ventricle. And then what we basically are looking at is the dorsal medulla. And what we see is the sort of end of the fourth ventricle with the obex. And we can see the gracile nuclei and the cuneate nuclei. And then what we basically see on the dorsal surface of the perichordal medulla is either the fasciculus gracilis running into the nucleus gracilis, or the fasciculus cuneatus running into the nucleus cuneatus. And then what we see also all through this is a series of the more laterally leading cranial nerves. So very often we end up using a very interesting term, or two terms. We talk about the spinal cord, and then we talk about the medulla, as if we were basically going and driving along some interstate in which you basically look and you see a sign and it says, you're driving through Eastern Ohio, and then all of a sudden you see a sign that says, Pennsylvania border 2 miles ahead. And you keep driving east and you go through the 2 miles, and what happened? And one part of the car might be in Ohio and the other part of the car is in Pennsylvania. But once you go from Ohio into Pennsylvania, you've gone into a totally different area. And very often when we have these arbitrary terms like the spinal cord or the cervical cord, and then we have the medulla, we basically think that the whole transition should be extremely abrupt. And the reality is that it is not. That is, there is a so-called spinomedullary transition zone. And we already encountered this when we were looking at the spinal sections, and we were looking at the upper and even as far back as the mid-cervical area. Why? What did we end up seeing? Even though we saw a clear dorsal horn and a ventral horn and a central canal, and we saw all of those spinal cord projection things, we all of a sudden also saw the emergence of the pyramidal decussations as they formed into the lateral corticospinal tract, actually doing that. So it basically tells you that this transition is an enormous number of fibers, enormous number of structures, and whatever. So in this, we should be on the outlook or lookout for 4 different things happening. Now, some of the things that are happening have incredible functional significance. But when you're looking at this grossly like we are, that functional significance is important, but you might not see that. On the other hand, there are things that we basically see physically changing. And even though those things that physically change are very clear and apparent, is it that important functionally? And the answer is no. Okay? So, in fact, if you look here, the first two of the spinal medullary transition is in the latter type of situation. So, what we basically look at here is we're going to be basically seeing, as we're moving caudally to rostrally, we're going to be basically looking at two structures in the spinal cord, the central canal and the dorsomedial septum. Okay? And what are those? Both of them are not very functionally important. They basically are spaces. The central canal, of course, contains the CSF as it runs through the spinal cord. The dorsomedial septum is actually an opening. It's a large sulcus. And what we know already, at least in the cervical cord, is that the posterior radicular arteries and the posterior spinal arteries would be sitting in the dorsomedial septum. But other than that, there's not that much functional significance. But what is basically going to happen? Where is the central canal typically in the spinal cord? It's almost like a bullseye in the center. It's almost as if you put a spinal cord section up and you play darts with it, and you would get a 10-point thing because you hit the central canal because you're hitting the center. Okay? So what is basically going to happen in the spinomedullary transition? The central canal is going to move more and more superiorly as we move rostrally. And then conversely, the dorsomedial septum, which is a thin sulcus, will now start to blossom and open like this, like a chevron, so that eventually what's going to happen is the central canal and the dorsomedial septum will merge and open into that horizontally aligned fourth ventricle. So now you figured out why, when I talked about the ventricular system, I talked about the orientation of the ventricular system, where we see the central canal in the periaqueductal gray sort of as tubular-like, whereas the fourth ventricle is like a chevron and it's horizontally aligned. So you basically see this happening, and it happens slowly. Now, when you're basically looking at the spinal cord, the one thing that is remarkably clear to you is that when you're looking at the sacral cord, the lumbar cord, the thoracic cord, and the cervical cord, we basically can see, in general, a similar arrangement of a sort of dorsal gray area, a ventral gray area, and an intermediate gray area. So there's a dorsal horn, there's a ventral horn, and there's an intermediate zone. And of course, the different levels of the spinal cord have different amounts of that, or they may have certain protuberances, like the lateral horn in the thoracic cord, or the nuclei of Clark, or things like that, or more alpha and gamma motor neurons in the cervical and lumbar versus the thoracic and spinal. But pretty much you have dorsal horn, intermediate zone, ventral horn. What now is going to happen? What's going to happen as we move rostrally into the caudal medulla is that distinction of dorsal horn and ventral horn will go away, and that intermediate area will now become what we call the medullary reticular formation. Okay? And of course, the structures that are within it, again, are very, very vague because, again, that word reticulum means net-like. And we're going to basically talk about that more. But now something of great functional significance happens. And we've begun to discuss this when we were talking about the cervical cord, and that is the pyramidal decussation. That what we know about the corticospinal tract as we move into the cortical medulla is that 95% of the fibers will decussate in the pyramidal decussation and go out and move laterally and superiorly to form the lateral spinothalamic tract in the spinal cord. And then 5% of the fibers will maintain that same trajectory and go into the cord as the anterior corticospinal tract and go through it. Okay? And of course, what's the major significance? The major significance is this is how major motor, integrative motor information is passing down into the spinal cord and passing down in a somatotopic fashion so that it innervates, differentially innervates the different levels of the cervical, thoracic, lumbar, and sacral cords. Then on the other hand, what we have seen is the so-called dorsal column system. And that when we finally get up into the C1 region of the upper cervical cord, we see very well established fasciculus gracilis, in which the medial portion of it is sacral fibers. Then the intermediate portion of the fasciculus gracilis is lumbar, and then lower thoracic. Then we have a dorsolateral septum, and then we see the cuneate fasciculi. And the more medial of the cuneate fasciculi are upper thoracic fibers, whereas then the vast majority of the cuneate area is what? Is cervical inputs. And what are the types of inputs? These are inputs, the vast majority of them are heavily myelinated fibers that have not synapsed in the cord and run through the gracial and cuneate fasciculi up into the dorsal medulla, where they synapse on the gracial and cuneate nuclei. And it's rather hard to see a synapse grossly, but we're going to basically start to see that. So these are the four different things that we are basically looking at. So when we look at the first spinomedullary section, we're going to identify the dorsomedial and dorsolateral septi with clear gracial and cuneate fasciculi. We're basically also going to see the central canal still there, even though we're in this spinomedullary transition. We're going to see the pyramidal decussation into the lateral corticospinal tract, and it literally interrupts dorsal gray matter. But then we're also going to see something interesting. That is, there are two cranial nerves that, although I defined them, the fifth cranial nerve, I said, would be found in the pons. The fifth cranial nerve, in reality, can extend as far rostrally as the midbrain and as far caudally as the cervical cord. So what we call the spinal fifth. We're going to see that. And when we look at these caudal exits, they are motor exits. Remember, the fifth cranial nerve is mixed. So it has a sensory component. Most of that is in the center or rostral portions of the fifth cranial nerve. And the motor portion is in the pons, but also in the medulla and also down in the spinal cord. And of course, the 11th cranial nerve, which we know is in the caudal medulla, also extends all the way down into the spinomedullary section, okay? And as we know, the 11th cranial nerve is purely motor, okay? So Here is a nice schematic of this. So what you're basically looking at here is to go through the major structures. There is your dorsomedial septum. There are your dorsolateral septi. So here you have the fasciculus gracilis. Here you have the fasciculus cuneatus. Here you have the dorsal horn. Here you have the intermediate zone. Here you have the ventral horn. So what do you see in the ventral horn in addition to alpha and gamma motor neurons? You are going to have the 11th cranial nerve, spinal 11th coming out laterally, and you see this spinal 5th coming out a little bit more medial to that. You still have a central canal. You still have a clear anterior medial septum. So if we look at this, here we are looking at what type of a stain. We are looking at a Wheel-Weigert stain. And a Wheel-Weigert stain, if you remember, is a fiber stain in which the Wheel-Weigert stains the myelin. So what you're seeing that's very dark is going to be white matter. So again, what do you see here? You see a very clear fasciculus gracilis. You see the fasciculus cuneatus. You see the dorsal horn. You see the ventral horn. You see the anterior median septum. And then what you see right here is literally the decussation of the pyramids. You can see some of the fibers are staying, but a large number of the fibers are literally decussating, going from the right side over to the left lateral corticospinal tract and the left corticospinal tract into the right lateral corticospinal tract. In the center here would be the central gray and the central canal. So this is a classic spinomedullary section because some of the things, the decussation of the pyramids has now happened. But everything else on it, the fact that there's still a central canal and dorsomedial septum, the fact there's still fasciculus gracilis and fasciculus cuneatus, and the fact that we still have very clear dorsal and ventral horns says that 3 of the categories are telling you that this is what? More spinal cord than medulla, okay? But we now move on. So now, an important thing to look at here is that the gracile, labeled by C, and the cuneate, labeled by D, fasciculi, are going to be synapsing on the gracile nucleus, or the gracile fasciculi, going to synapse on the gracile nuclei that we're going to be labeled as B. Now, the important thing is, in this very caudal medullary section, we don't see the cuneate nucleus much because that starts to appear a little bit more rostrally. And we'll see this in the succeeding sections. So now, what you basically, again, are going to continue to see is that the dorsal horn labeled by F and the ventral horn labeled by I is going to be interrupted by these decussating pyramidal motor fibers, L, moving towards the lateral corticospinal tract, okay? And then we're also going to see, labeled in this section, the central canal. So here we go. Here, again, is a Wheel-Weigert stain. And what you can basically see here is a very nice dorsomedial septum. You see the dorsolateral septi. And then labeled by the letter C is the fasciculus gracilis. But now, unlike when we look back here, where we just saw just plain old fasciculus gracilis all the time and plain old fasciculus cuneatus all the time, when we look in this section, what do we see right here in the ventral portion of where the fasciculus gracilis should be? We basically see the letter B, and what we don't see is a stain that's picking up the myelin. Why? Because right here in B is actually cells, and what are they? They're the cells of the nucleus gracilis. So the fibers of the fasciculus gracilis are now synapsing on the nucleus gracilis. And of course, in this dorsal column system, this is the very first synapse for those A alpha and A beta fibers that entered, remember, all the way down in the sacral cord, or they entered in the lumbar cord, or they entered in the lower thoracic cord. They are now starting to synapse in the nucleus gracilis, synapse number 1. And of course, there are dorsal column postsynaptic fibers, a minority, but they're coming in from what? Laminae through 2 through 5, and there's second order neurons that go in, and they are synapsing here. And a very large, again, 2 through 5 can synapse here. Notice with D, when we're looking at the fasciculus cuneatus, we see very little evidence of the accompanying cells of the nucleus cuneatus, because that's happening more rostrally. Then what we again see is the structure that we always are looking for, the dorsal horn and the ventral horn. And then what we basically still continue to see is this continuing decussation of the corticospinal tract, so that the fibers are eventually going to move out into what? The lateral corticospinal tract. And again, what you see is now a slightly wider opening of our friend, the dorsal medial septum, but we still see the central canal sort of in the bullseye, right in that center of the spinomedullary structure. So now, when we continue to move up, we're basically again saying that the pyramids are decussating. 95% of them are decussating, 5% is not. And that again, we continue to see the synapsing of that massive dorsal column system of the gracile and cuneate fasciculi into the gracile and cuneate nuclei, and is again going to be more gracile nuclei synapsing in the caudal extent. And we still see spinal fifth and 11 cranial nerves, but now we're starting to see that the spinal gray, that very distinct dorsal versus ventral horn, is now getting more ambiguous, and we start to call that a reticular gray. So here is a schematic drawing of this. So again, you still see our friend, the central canal, sitting in the dead center. We still have dorsal medial and dorsal lateral septi. We still see a fasciculus gracilis and a fasciculus cuneatus, fasciculus gracilis and fasciculus cuneatus. And now these sort of bunny ears that you're looking at right here is still now seeing the increased number of gracile nuclei cells, where there is this great amount of synapsing. And then what we also see here is now in this slightly rostral section is, in addition to this robust fasciculus cuneatus, you're now starting to see the emerging nucleus cuneatus, okay? Now, what else are we starting to see? Notice that we don't necessarily call this the dorsal horn anymore. That actually, when we're looking way out here, we're actually looking at the beginning of what we would call the inferior cerebellar peduncle. The inferior cerebellar peduncle here and the inferior cerebellar peduncle here, which the other name for that is the restiform body. And where is it sitting? It's sitting right in the trajectory of where the posterior spinocerebellar tract is. So this is where, again, we can use that wonderful word comprised of, that the inferior cerebellar peduncle is going to be comprised of, in part, by the posterior spinocerebellar tract. Okay? So now, like I said, we don't see a clear dorsal horn and ventral horn. It's getting sort of vague. So we have this sort of very caudal medullary reticular formation, but we still have a pericanular gray, and we're still going to have, that lamina 10, and we have the central canal. Now what we can see very clearly is not, remember in these previous sections, when we looked at here and we look here, we see the lateral corticospinal tracts already decussated. Here we're seeing part of the decussation, but we're seeing them decussating. Here you're really seeing both the original corticospinal tract in its original position and as they decussate. And then, of course, you see spinal 11th exit and spinal 5th exit. So now when we look at that, this is what it translates out to. So now let's, again, the whole point of this is, as you're looking at sections, you're asking what stays the same, then what was there before that no longer is, or how is it substantially changed, and then what is something that's new in the section. That's how you should approach this. So if we go all the way back to here, what do we basically see when we look at the fasciculus gracilis? All we see is fasciculus gracilis. All we see is fasciculus cuneatus. Here, we see quite a bit of fasciculus gracilis, and we see total fasciculus cuneatus, but what do we see? The emerging nucleus gracilis. Not much nucleus cuneatus. Now, in this section, what are we now seeing that occupies the center? What we see is very little fasciculus gracilis and mostly nucleus gracilis. Here we see quite a bit of fasciculus cuneatus, but we see an emerging nucleus cuneatus. What you still see right here is a little bit of dorsal horn and ventral horn, but what you're basically seeing a lot of is the dam decussation of those huge pyramids, and we're actually looking on the inferior surface of the brain, and versus before, where we looked at this and we can basically say, here's the anterior white commissure, not the anterior medial septum, and here's the anterior corticospinal. Here you're starting to see some pyramids on the bottom. But here we're seeing definite pyramid on the bottom. So we can see we're moving nostrally, yet we still see our central canal, and we still see our posterior medial septum. Okay. So now, we want to again continue to see the Gracile B and cuneate D fasciculi synapse on the Gracile and cuneate nuclei marked by C and E. Then, now that we see the Gracile and cuneate nuclei, we know that they give rise to an enormous number of fibers that are first going to form the internal arcuate system and then eventually form the medial lemniscus on their way all the way up to the ventral-posterolateral nucleus of thalamus. So, in addition to seeing the Gracile and cuneate inputs into the Gracile C and cuneate E nuclei, we are now going to look at the outputs of the Gracile and cuneate nuclei to form these internal arcuate fibers, M, that will course around the pericanular gray central canal and then decussate below the central canal to form the medial lemniscus that we call N. We have moved rostrally. So now what we're going to realize is that the corticospinal tract, which decussates caudal to us, is not decussating. But you still can see some small remnants of dorsal and ventral horns. So let's look at that. So now look at this. Look at how little of the fasciculus gracilis there still is. There's still some remnants of it, because it'll keep synapsing rostrally. But the enormous number of Gracile fasciculus neurons have already synapsed to form this very, very large now nucleus gracilis. And then what I basically think of is looking at like a tooth here, D, which is the cuneate fasciculus. It sort of surrounds the pulp, E, of the cuneate nuclei. So you see them there and there. So now we pay attention to the axons of the nucleus gracilis and the axons of the nucleus cuneatus. They course inferiorly, and remember right here is the central canal and a pericanular gray, and notice where the central canal is. It's slowly starting to move dorsally, and notice our dorsal medial septum is starting to open up. Eventually it's going to form a fourth ventricle. But what's happening? These Gracile and cuneate nucleus fibers course around the central canal and pericanular gray and go around that, and you can see M all the way around here and M all the way around here. They come and they decussate right under the central canal to form the medial lemniscus. So the medial lemniscus is the fibers of the Gracile and cuneate nucleus. This particular N is coming from these cells. This particular N is coming from these cells because the internal arcuate fibers have decussated. Then what we basically can still see here is some small remnant of a dorsal horn and a little bit of a ventral horn. But what you have in addition is a whole reticular gray. Then we pay attention to the inferior surface of the brain here. What you can basically see now is a huge corticospinal tract. So in the pyramids, so the pyramidal tract K is from the ipsilateral side of the precentral gyrus. This is from the other ipsilateral side. Okay, so we just started to see all of these things happening. So now we have a caudal medullary section. We have a nucleus gracilis and nucleus cuneatus, and the fibers form the internal arcuate system and decussate to form the medial lemniscus. And in this caudal medullary section, that medial lemniscus is going to be organized in a vertical orientation. Now, when we look at the nucleus gracilis, actually, when we look at the nucleus cuneatus in this more rostral section, we're actually going to see two nuclei. We're going to see the main cuneate nucleus, which we referred to up here, and the accessory cuneate nucleus. And the accessory cuneate nucleus that basically is going to look like a little duckbill on a duckbill head, you'll see that in a second, are actually a different type of input. Remember that when we saw the posterior and anterior spinocerebellar tracts, where did they come from? Where they came from was the nuclei of Clark, which was found somewhere around L1, L2, and all the way up to C8, but not rostral to C8. So the nuclei of Clark supplied to the cerebellum via the anterior and posterior spinocerebellar tracts, sacral and lumbar and thoracic input into the cerebellum. There is no cervical input into the cerebellum via that pathway. Where does the cervical and upper thoracic inputs to the cerebellum come? They come through the accessory cuneate nucleus. So this completes spinal input to the cerebellum. The posterior spinocerebellar tract supplies lower thoracic and lumbar input. The accessory cuneate nucleus and the cuneocerebellar tract is supplying cervical and upper thoracic cerebellar input. So now, let's look at that. So what we have here, let's start to notice some things that have remained the same and some things that are now different. So one thing that's remained the same is that we now can see the corticospinal tract has not yet decussated, and in that caudal section we didn't see it. We had to go more caudally in order to see that decussation. What we also start to see is we still see the vertical orientation of the medial lemniscus here and here. So what I basically say here, again, I can think of this as a Rorschach test, and what I see is two little Christmas stockings with eggs sticking out here and here, okay? So what you have, this sort of little Christmas stocking that I basically see here is the medial lemniscus, and that's keeping its organization. However, remember we were looking for the dorsal horn and the ventral horn turning into a reticular gray. And what we no longer see is any dorsal or ventral horn. We see reticular gray, and then we see some very interesting distinctive structures, okay? Then we looked at the fasciculus gracilis and the fasciculus cuneatus synapsing onto the nucleus gracilis and nucleus cuneatus. And of course, we see this very, very strongly here with the nucleus gracilis here and the nucleus cuneatus here. And then what we see, if we can notice two duck heads looking out laterally, we see a nice little duck bill, and that duck bill is the accessory cuneate nucleus. This duck bill is the accessory cuneate nucleus. So now, the axons of the accessory cuneate nucleus is going to form the cuneo-cerebellar tract. It's going to form the cuneo-cerebellar tract. So what do we have to do? We have to get that cellular input up into the cerebellum. What's the easiest way to do this? Is that the cuneo-cerebellar tract enters into the inferior cerebellar peduncle. The cuneo-cerebellar tract enters into the inferior cerebellar peduncle. So now let's go through that word comprising again. What we have now learned is that the inferior cerebellar peduncle is comprised in part by the posterior spinocerebellar tract and the cuneo-cerebellar tract. So what you can see is that input from the lumbar, from the thoracic, and from the cervical cord is now within our friend, the inferior cerebellar peduncle. Okay? So now, what is the fourth thing we've been paying attention to? The fourth thing we've been paying attention to is the dorsomedial septum and the central canal. Notice the central canal, if it was in the center, should be right here. What has now happened is we've moved rostrally. The central canal is drifting superiorly. What also is happening? The dorsomedial septum is widening. So eventually this is going to form the fourth ventricle. It hasn't yet. But if there was a fourth ventricle, and we're going to see the fourth ventricle, we would expect to see things around what we call the periventricular gray. And one of the things that we're going to notice in the medulla, in the periventricular gray, is this is where a number of our cranial nerve nuclei, not all of them, but a number of our cranial nerve nuclei are going to be found. Now, remember how we're looking at things. We're looking at things moving caudally to rostrally. Yet, when I define the cranial nerves for you, I called them cranial nerves 1 through 12. But the way the cranial nerves are numbered is moving rostrally to caudally. So, if we're detectives here, and this is the first time, other than the spinal fifth and other than the spinal 11th, we're going to see the central structure of the cranial nerves. We would be looking for cranial nerves 12, 11, and 10. So now, let's think of the 12th cranial nerve. The 12th cranial nerve is the hypoglossal. We defined it as being in the medulla, and given that number 12 is the most caudal. Okay? That's good so far. What did I tell you about the hypoglossal nerve? I told you that that was a motor nerve. So it is an efferent. The fibers that are coming out of the 12th cranial nerve are controlling the posterior 2/3 of the tongue. So therefore, its major role is an incredible role that we don't necessarily pay attention to unless all of a sudden we do something like me gulping coffee. That is the process of swallowing, that you utilize the posterior 2/3 of the tongue to take, when you swallow, to move whatever food is in the mouth or liquid in the mouth and have it swallowed. So one of the things here that we can see is when we're looking at the cranial nerves, we have two designations. One is a dot followed by a fiber, and the other is a fiber followed by what looks like, I don't know, a depiction of a snake, the forked tongue of a snake. Well, what the dot is showing is demonstrating that it is motor, so that the 12th cranial nerve, the nucleus, is found here, and it is sending out a motor efferent. Now, remember I told you had to learn that the 12th cranial nerve is the hypoglossal. It's controlling the posterior 2/3 of the tongue. It's found in the caudal mesencephalon, myelencephalon, and it is exiting the brain medially. And you might have been basically saying, when I told you to learn that stuff, is that, Jesus, that's just one piece of too much information. Now I hope you're going to recognize why I'm teaching you that. It's because when we see these different cranial nerves, and I'll identify the others in a minute, that we see the hypoglossal, as it exits the brain, is exiting the brain more medially than the other cranial nerves that are exiting the brain more laterally, okay? So the 12th cranial nerve, hypoglossal, in the caudal mesencephalon, is exiting the brain medially and controlling the posterior 2/3 of the tongue. So now, I know we're not in the periventricular gray yet, but you can see the most medial portion of the periventricular gray in this caudal medulla is the hypoglossal. So what are two cranial nerves that are so-called lateral to it? Well, right here is two components of one cranial nerve, and that is the 10th cranial nerve, the vagus. And we learned that the vagus is the largest of the cranial nerves, and it is going to innervate the sort of whole internal viscera. And we divide the vagus into a supradiaphragmatic vagus, which is primarily controlling the lungs and the heart, and therefore controlling respiration and some type of cardiac control. And then the subdiaphragmatic vagus, which is controlling the whole digestive viscera and controlling digestion. It is a mixed cranial nerve. So what does that mean? There's a motor component and that there's a sensory component. Now, even though when we look at this section and when we think of the fourth ventricle as being horizontally aligned, we should basically think of where would these where would this mixed cranial nerve be? Well, there's a rule for all of the mixed cranial nerves. And which ones are there? There is the 5th cranial nerve, there's the 7th cranial nerve, there's the 9th cranial nerve, and there's the 10th cranial nerve, okay? They both have sensory afferents and motor efferents. The motor portion of the 10th cranial nerve is found, let me make the general statement. The motor portion of a mixed cranial nerve is going to be medial to the sensory portion, which is going to be lateral. So even though we haven't gotten the fourth ventricle yet, the motor 10th is medial to the sensory 10th. Okay? The motor 10th is medial to the sensory tent. Okay? So what is that motor tent doing? That motor tent is controlling the supradiaphragmatic vagus, respiration, and cardiovascular, and subdiaphragmatic vagus, the digestive and excretory systems, and whatever the motor tent is controlling that. Then conversely, the sensory tent is bringing in somatosensory, viscerosensory, and interoceptive types of information into there through the sensory branch of the vagus nerve. And then when we talked about the vagus nerve, we pointed out it was large, and we pointed out that it enters the brain and exits the brain laterally. And that it obviously, when it's entering and exiting the brain, it's going through what we call the cranial nerve foramina, the openings in the skull, so that those fibers can go in and out. Now, one other thing we learned, and it was a piece of minutiae, is that whereas the 10th cranial nerve is the largest, its partner, the 11th cranial nerve, is the smallest, the spinal accessory. And then I used that old term of saying, with Rodney Dangerfield saying, I can't get no respect, that I'm the smallest cranial nerve, and I'm so goddamn small, I have to share the same foramina to exit with the 10th. So there is the 11th cranial nerve. Now notice where the 11th cranial nerve is located. Is it located right around the periventricular gray? And the answer is no. It's located out here in what we would used to call the sort of intermediate zone of the spinal cord. But since we're no longer in the spinal cord, what are we in? We're in this vague structure called the reticular formation, okay? And so there is a nucleus sitting within the reticular formation. Now, to complete my whole story of how disrespectful we are to the 11th cranial nerve, folks gave the 11th cranial nerve nucleus a name. And the disrespectful name it gave it was the nucleus ambiguus. So the nucleus ambiguus is the 11th cranial nerve nucleus, and it is found in a vague area called the reticular formation, and it shares the same foramina as the 10th to exit the brain, and then it controls what we call the branchiomeric arch and sort of the full expansion of the limits of what we can do in terms of inspiration and expiration. Okay. Let me just look here for a second. I'm just stopping the share here for a second. Good. I just want to see where I am with time. I'm still going strong. Good. So now we have just identified the reticular formation. We identified the periventricular gray. And now what's happening with the fibers that are the second order fibers that are coming out of the nucleus gracilis and nucleus cuneatus, they're still forming internal arcuate fibers to form the medial lemniscus. So now we have two other structures that we want to pay attention to. One structure is, if you've noticed, is that the medial lemniscus, which literally was totally adjacent to each other on the left and right side, are now separated. And a nucleus starts to emerge right here in that separation. And that nucleus is called the raphe nucleus, R-A-P-H-E with the accent grave on it, the raphe nuclei. Now, depending on the species that we study, we can have anywhere from 9 to 12 raphe nuclei, okay? And the word raphe is very interesting because look at where it is. It sits right along the midline or the most medial portion of the section. So literally, if we were to take this section and fold it in half on top of each other, laterally and medially, and fold this way, where would the raphe end up being? It would end up being right in the fold or the slit. And the word raphe roughly translates into fold or slit. Now, I'm pointing out this nucleus, and that there's 12 of them, and here's one of the most caudal raphe nuclei. These raphe nuclei are always found right along the midline, anywhere from the caudal medulla all the way up into the rostral midbrain. And other than the fact that they were there and could be identified cytoarchitecturally, that made them an interesting structure. Until 1964. And in 1964, there was the first in a series of incredibly influential studies in neuroanatomy that revolutionized or integrated the anatomical underpinnings of the brain with the brain's emerging neurochemistry. In 1964, Annika Dahlström and Kjell Fuchse, F-U-X-E, Dahlström and Fuchse, in 1964. Then in 1971, another Swede by the name of Urban Ungersted. And then in 1976, two other Swedes, Ali Lindvall and Anders Björklund, basically went and did a series of studies that literally identified some of the neurochemical makeup of these neuroanatomical structures. And the raphe were one of the structures. What they basically did in the initial series of studies is use an anatomical process called histofluorescence. That is, and this was discovered much further, that there was lots and lots of fluorescent types of materials. Folks have won the Nobel Prize for this and whatever. But in histofluorescence, what you basically do is you take a brain section and then expose it in under liquid nitrogen to a paraformaldehyde type of solution. And for a brief period of time, certain cells will fluoresce. Some of the cells fluoresced in a deep green type, lime green type of color. And what was discovered is that those cells that fluoresced, that lime green color, were cells that contained catecholamines. And the two major catecholamines that we study within the central nervous system is the neurotransmitters norepinephrine and dopamine. Also, you get epinephrine also fluorescing peripherally, and there's a small amount of epinephrine in the brain. So literally, when you did this histofluorescence procedure, followed by another procedure called glyoxylic acid staining, you could basically identify the cells in the brain that contained norepinephrine and dopamine. At the same time, what they found is that there would be cells that contained another very interesting structure. And that is a related substance called an indolamine. And indolamines give rise to another group of active neurotransmitters, like melatonin, but especially the neurotransmitter serotonin. And what was basically found here by the Dahlstrom and Fuchs lab, then Ungerstedt, and then Lindvall and Jorkland, is that the raphe nuclei, right here, fluoresced and basically showed us that it contained serotonin as a transmitter. That over the years, it basically has been found that about 1/3 of the cells that can be cytoarchitecturally defined as raphe nuclei also contain the neurotransmitter serotonin. Now, so what we have with the advent of histofluorescence and then glyoxylic acid is the beginning of what we called neurochemical neuroanatomy, okay? And that this neurochemical neuroanatomy in these early studies basically identified a group of catecholamine cells that stained lime green and a group of indolamine cells with serotonin that stained this dull yellow. And of course, this all happened between 1964 and 1976. And what also started to happen in a period just before that, in a period from the 1950s into the 1960s, there was also another revolution in psychiatry. And the revolution in psychiatry was the beginning and strong use of psychopharmacological types of drugs. And that a number of the early drugs that were used, the amphetamines, the psychotropic drugs, and antihistaminergic drugs that turned into antidopaminergic drugs, of course, were things like chlorpromazine and thorazine and haloperidol and everything that were acting on noradrenergic and dopaminergic synapses between the amphetamines and whatever, and then that would excite these systems, and then the thorazine and haloperidol that would block these receptors. But also at the same time, there was the beginning of the development of antidepressant drugs. And one powerful and early antidepressant drug that was found was a drug called imipramine in the late 50s and early 60s. And eventually this was found to do what? Was to modulate serotonin transmission. So now, with the advent of histofluorescence and visualizing catecholamines and indolamines, one could now visualize and study exactly where the neuroanatomical structures in the brain were, and that what you're basically looking at here is in the very caudal medulla, we have a serotonergic pathway. One more divergence that comes out of the our friend the serotonergic pathway in the raphe is, I told you that 1/3 of the cells are serotonergic. 2/3 of the cells contain other transmitters. Now, roughly around the time of the 20s and 30s, when people found serotonin and norepinephrine and dopamine. A very powerful neuroscientist at the time, the British scientist Sir Henry Dale, came up with a law that basically said that, yes, there are neurons that contain these neurotransmitters, and that any given neuron that contains the neurotransmitter releases that and only that neurotransmitter. So a serotonin cell can't be a dopamine cell, can't be a norepinephrine cell, can't be an acetylcholine cell. It's only a serotonin cell. Well, right in the mid-70s, with the fact that people finally discovered what a major other drug class bound to in the brain, and that's the opiates, they found that there was an opiate receptor. So therefore, if there was an opiate receptor, there were endogenous opiates, just like a dopamine receptor is being bound by dopamine release from another cell. A noradrenergic receptor is being bound by a norepinephrine containing cell releasing. So in the opiate receptor, there should be endogenous opiate pathway that releases endogenous opiates. And of course, the great shock from between 1973 and late 1975, there was literally a search that, again, when I go into sort of descriptions of it, we can basically think of the wonderful fairy tale Cinderella, that all of a sudden, the prince is at the ball and this young woman comes in and he dances with her, and then all of a sudden the bell starts to ring and Cinderella has to run off because at midnight all of her clothes would change and her carriage would change into a pumpkin. Great story. And of course, the next day and the next weeks, what does the prince do? He has the golden, the slipper, and he goes through the kingdom and he tries to slip her on all of the young ladies of the realm. And he finally finds Cinderella and he sees the slipper fit, and poof, we have absolute love. So what are we basically looking at there? We're looking at how an endogenous substance that's produced by one neuron that's released binds to the receptor, the opiate receptor. And of course, what was the great shock? The great shock is when they found the first endogenous opioids. They were not like acetylcholine or norepinephrine or dopamine or serotonin. What do those four things have in common? What they have in common is they are derived from a single amino acid that undergoes enzymatic changes, okay? And they were not like the other two types of transmitters that were now understood, the so-called excitatory neurotransmitters, as demonstrated by glutamate, and the inhibitory neurotransmitters that were demonstrated by GABA. There was something different. What they were, the first two that were discovered by using constellates, were pentapeptides. They were strings of amino acids, 5 amino acids strung together to form a peptide that had this incredibly high affinity for an opiate receptor. Within a few months, there were other peptides that were discovered: beta endorphin, dynorphin. Decades later, nociceptin, endomorphin. All of them were polypeptides. Now, people knew there were peptides all around the brain, and they gave them no thought. Now, all of a sudden, it was fair game. So when I went to graduate school, and I graduated within 4 years, in late 75, early 76, what I was responsible for was about 8 known neurotransmitters. And I had to figure them out. by the time you got from late 1975 up to 1977, 78, there were now 100, 200, you could literally talk about 1,000 neurotransmitters that are really put into all different kinds of categories. That we have neurotransmitters that are single amino acids that undergo enzymatic change. There's the excitatory and inhibitory amino acids. And then there's that global thing of what do you call it, peptides. And a new technique came along when we started to look at peptides that then was also used for the study of classic neurotransmitters. And that technique was called immunocytochemistry. And in immunocytochemistry, what you're basically using are antibodies to the particular substance that you're studying. So in the case of a peptide, let's say there is beta endorphin, you can create an antibody to beta endorphin. Or if you want to study immunocytochemistry with classic neurotransmitters, what you don't do is create an antibody to norepinephrine. You can't do that. It's not very good. What you create is an antibody to the rate-limiting enzyme. In the case of catecholamines, you would either use tyrosine hydroxylase or dopamine beta hydroxylase, an antibody. And then, of course, you're going to create at the beginning polyclonal antibodies in which they would have rabbits, draining the things out of the ears to get an antibody titer, or eventually going into monoclonal antibodies, where you literally get the thing with a much straighter thing. So now those Swedes keep studying, except now another guy enters the fray, a man by the name of Thomas Hochfeld, H-O-K-F-E-L-T, and a whole bunch of other people, but his lab probably in about a 4-year period published something like 70 to 100 papers that basically studied the immunocytochemistry of peptides. So if I go back to our friend here in the caudal raphe, what you basically find is the caudal raphe nuclei. Remember, 1/3 of the cells are serotonergic. A load of the cells are peptidergic. So peptides like thyrotropin releasing hormone, TRH, or substance P can be found in the caudal raphe. So you go, well, that's not surprising. You told me 2/3 of the cells don't contain serotonin. So what? So some of them contain substance P, some of them contain TRH. You still can follow Dale's law. But what does Hochfeld discover? What he basically discovers that some of the cells, subpopulations of some of the cells that are immunopositive for serotonin are also immunopositive for substance P. And then some of the cells are immunopositive for serotonin, and TRH. And then even some of the cells are immunopositive for serotonin, substance P, and TRH. So all of a sudden, good old Dale's law goes right out the window, because you now find neurons that can release a multiplicity of substances. What they don't do is release, so you don't find a cell that releases both glutamate and GABA. They release either one. You don't find a cell that releases norepinephrine and dopamine, but you can find a cell that releases dopamine that also releases cholecystokinin. You can find a cell that releases serotonin, but also releases substance P and TRH. Now, what's very important about that is that when you basically study how these cells release it, you know the classic thing of release. Basically, you're talking about vesicular release, etc., and you release packets of the neurotransmitter. That's for the classic neurotransmitter, like the indolamines, like the catecholamines, and cholinergic drugs. Peptides don't get released that way, which basically means that when there's an action potential in the caudal raphe and you release serotonin in one way, you release TRH and substance P in another way. And the other point is that when you release something, what else basically is happening here? We're putting a lot of things together. That when you look at the classic neurotransmitters, like norepinephrine, dopamine, serotonin, acetylcholine, histamine, etc., what are they basically doing? That there's release, there's binding to a receptor, but then what basically happens is there's this other process called reuptake through all kinds of transport mechanisms. So what's the consequence of this? And also all happens in a Bhutan. What's the consequence of all of this? That if I shoot my gun one time and I release my serotonin out into the synapse, 95% of that serotonin can get reuptaken for resynthesis and shooting my gun again. But yet at the same time, if I decide to shoot a gun and I release my serotonin, I also release my TRH and substance P, but my substance P and TRH can never be reuptaken. I have to produce that much more. So there's a whole bunch of molecular processes going on here that you have to begin to understand about the underlying things about these cells. But here, the most caudal, and we'll be seeing them as we march through the medulla, is the raphe nuclei. Then, of course, we see the corticospinal tract. Then we have the reticular formation. Now, let me just check again to see where I am. Okay, I will finish this slide today. Good. That was my plan. I spend a lot of time on some slides because there's a lot of new things that are happening here. So here we go. So as I pointed out, the medial lemniscus is this sort of Christmas stocking. Now let's look at this little bump right above the Christmas stocking, like you got either a piece of coal or an egg in your Christmas stocking. And what you have there is another fiber tract. It is called the medial longitudinal fasciculus, or MLF. Okay, so what I'm going to do is do something with the MLF. Okay, I know I can't force you to do this, but I have everybody, I'm looking at everybody. What I want is everybody, if you can, to turn on your video. Please turn on your video because I want you to do this activity, okay? And try to get yourself centered in the center of the video so everybody else can see what everybody else is doing, okay? All right, now, just imagine that what you had, put your finger up right by your nose and bring it out like this, okay? Now, what I want you to do is move your finger to the left and then, without moving your head, move your eyes. Now move it to the right, move it to the left, move it to the right, without moving your head. Which way are your eyes moving? They're moving medially, laterally, to the left and the right. Okay, now I know it's 11:45. I've been screaming at you for the last 2.5 hours. And if I tell you to do this, you may fall asleep. Put your head on your right shoulder as much as you can, like that, okay? Everybody put their head on their right shoulder, okay? Very interesting. I go that way, you all go that way, okay? And again, put that finger up. And now move the finger to the left and the right. And follow it with your eyes. Which way are your eyes moving? They're moving up and down. Everybody recognize that? Good. Okay. So now, what is this medial longitudinal fasciculus? Let me get back to this. This medial longitudinal fasciculus is a fiber tract that is a bifurcating axon. So part of the fibers go rostrally and part of the fibers go caudally, okay? They go as far caudal as the caudal medulla, because you can see the MLF right here, right? And these fibers can go also as far rostrally as the middle of the midbrain. Now, think for a second. What's in the middle of the midbrain? The third cranial nerve, the oculomotor nerve. And the oculomotor nerve controls a whole bunch of eye muscles that we're going to go over. And then as we move through the caudal midbrain, we pass the fourth cranial nerve, which controls another set of eye muscles. Then we go through the pons and we get to the medullary pontine border, and we're at the sixth cranial nerve, which controls yet another eye muscle. And then as we move through the medulla, we are now moving through things where there's the vagus nerve, where there's the glossopharyngeal nerve, where there is the facial nerve, and then rostrally the trigeminal nerve, which is giving us what? All types of somatosensory information about what? The position of our head, the position of our neck, the position of our body. So all of this information is coming in, but there's also the motor information as to where we are in space, and the MLF is sending out this information. Why? Where does the MLF originate? It originates in the rostral medulla in a place called the medial vestibular nuclei, which is getting what? Information from the vestibular cochlear nerve that basically tells us where our head, where our shoulders, where our neck is in space. So we now get that information, that medial vestibular nucleus information, sent out via the MLF, that then informs all of the other motor nuclei to basically, if you're doing tracking and things like that. And that basically is happening with the MLF. So now the last structure, and again, when I saw it the very first time, I said, oh, gee, this is a case of trichinosis, worms on the brain. That you had this very, very strange set of structures in the caudal medulla. What you have here is this structure right here. And then you have this structure and that structure. Now, what I want you to do is do a thought experiment. Think when you went back to gross anatomy, and we looked at the inferior surface of the brain, and we looked at the medulla. And what we saw were two medial bumps, right? We saw what we identified as the pyramids, And then we saw two lateral bumps out here that we identified when we looked at them as the olives. They basically looked to old Roman neuroanatomists like something that they would eat off of a tree, an olive tree. So, quite logically, this set of nuclei right here is called the inferior olivary complex. The inferior olivary complex. Okay? And the inferior olivary complex is comprised of three major nuclei. The principal nucleus of the inferior olivary complex, the dorsal accessory nucleus of the inferior olivary complex, and the medial accessory nucleus of the inferior olivary complex. So the inferior olivary complex is made up of the principal nucleus, the dorsal accessory, and the medial accessory. Now, the inferior olivary complex, the principal nucleus, has what you basically call a horseshoe type of shape. The area inside the horseshoe, which is just reticular formation, is given a name because it has a sort of an interesting look. It's called the hilus, H-I-L-U-S. So now, what is the inferior olivary complex and why is that important? Well, one thing, we're going to finish that whole idea of comprising things. There are two major types of fibers that leave the olivary complex and they travel up to the cerebellum. Okay, two types of fibers. So there's two types of what we call olivo-cerebellar fibers. There are going to be climbing fibers and they're going to be morsy fibers. Literally, it goes back to baseball and Wrigley Field, where when you basically look at Wrigley Field, and if you ever watch a baseball game, and I think you can watch the Cubs play today, they may blow it yet again against the Miami Marlins. But if you go in the inside of Wrigley Field, you have what are called the ivy, that is climbing ivy. And literally, the people who studied olivocerebellar fibers and recognized that there were two types of them. You literally use the term climbing fibers and then morsy fibers that climb their way up to the cerebellum. Now, how do they get up to the cerebellum? Well, look at where the principal nucleus is and the medial and dorsal accessory nuclei are. They utilize our good old friend, the inferior cerebellar peduncle. So now I can close the loop. Yes, there are other fiber tracts in the inferior cerebellar peduncle, but what you should be interested in is the three major types of inputs that are going through the inferior cerebellar peduncle to the cerebellum. There's the posterior spinocerebellar tract, there's the cuneo-cerebellar tract, and now we have the olivocerebellar tract. Now, whereas the posterior spinocerebellar tract and the cuneo-cerebellar tract give inputs from different levels of the spinal cord, with the posterior spinocerebellar giving lumbar and thoracic inputs, and the cuneo-cerebellar giving cervical input. The olivocerebellar tract is giving input from the brain that is now processed. Because if you remember, I spent a bit of time at the beginning of the lecture on two major output pathways. One was our friend, the Corticospinal Tract, and the second that goes all through here was called the Corticobulbar Tract. So the Corticobulbar Tract, which is what? Information that's already been processed by the brain and going out again, will modulate further input that is coming into climbing fibers and morsy fibers. So let me see where I am so I can know where I should stop. Okay, so now let me go very quickly, and we're going to go through this again in detail. Climbing fibers. What climbing fibers do is they climb up into the cerebellum, all the way up into the cerebellar cortex, and they end up synapsing on Purkinje cells. And they synapse on these Purkinje cells in the cerebellum in a highly specific manner. And when they synapse on the Purkinje cells, they excite the Purkinje cells. Morsy fibers, on the other hand, climb up into the cerebellum and go into layer 3 of the cerebellum, where they then synapse on cells in the layer 3 that then sends fibers back up to layer 1 of the cerebellum, so-called parallel fibers. And these parallel fibers, which are excited by the morsy cells, go and inhibit Purkinje cells. So now, what the hell does that mean? If you go back to our friend, the pyramidal cell in the precentral gyrus, the pyramidal cell, when it fires, is going to excite something. The Purkinje cell, when it fires, is going to inhibit something. So, a Purkinje cell, how many layers in the cerebellum? You'll learn 3. By the way, you can always Google it. Okay, there's 3 layers. Okay, versus 6 layers in the cortex. But so the Purkinje cell, which basically inhibits the ability of the Purkinje cell to inhibit, is enhanced by what? Climbing fiber excitation. You're exciting an inhibitor, producing greater inhibition. On the other hand, a Purkinje cell is inhibited by morsy fibers. So what you're doing is you're inhibiting an inhibitor, which means what? You're disinhibiting, which is another way of saying you're allowing something to be excited. So basically, if you think of the cerebellum, and you always can go and think about it, again, wonderful movie to basically go and see, watch just one part of it, one of the earliest flicks by Woody Allen. In Woody Allen, you had the movie, Everything You Wanted to Know About Sex. Okay, and they had all these actors doing various things. And when he gets into the nervous system, and then, of course, the two things are the funny thing when he gets into the nervous system, and there is the so-called part of the nervous system, remember, he's very Freudian, he's psychoanalytic, that's basically all id. Do it, do it, do it. And he hears, I'm sorry, I'm going to be politically incorrect here and terrible and sexist. But in the movie, I'm just quoting Allen, is, the guy just met a co-ed from NYU. God, I guess for Woody Allen, that meant something. But in any way, and all of a sudden you see everybody jumping up and down, getting excited and everything. And then, of course, there's another part of the thing, which is the superego going, no, 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 no. And of course, then you then ship down into the sperm, and you have guys like Burt Reynolds and everything like that being a sperm cell. It's a hilarious movie. Go watch that. But anyway, if you're basically going and thinking about, am I going to reach for this this coffee cup or not? Then bring it to my lips, sip it, swallow it, and now start talking without choking and all of that. You have this great degree of interplay. It's like, wait, stop, now, okay? And the way you're doing that is not only do you have the excitation of all of your motor pathways, but you have this modulation of the cerebellum through the Purkinje layer by either saying no, and then finally something breaking through where you go and you say yes, and do it now, and do it very specifically, okay? So like last night, I did not stay up all night for the Yankee game. In fact, what I did is, being a 74-year-old man who underwent prostate surgery about a year and a half ago, I'm very happy to say I only wake up one time during the night to help myself, okay? And of course, what I did was I went to bed at about 11:30 last night. The Yankee game, it was 6-6 and everything. And now I wake up and I look at the clock and it says 4:30 in the morning. And I get out of bed, I help myself, and I walk all the way downstairs, turn on the computer, and find out that the Yankees won 10-9, okay? And the point is that if you watch the replays of Gio Urshela hitting a grand slam home run that gave the Yankees a brief lead and it went back and forth for the whole game, what you watched was the pitcher. And by the way, the pitcher, if you go and look, just as I'm a Yankee fan, I always sit there and laugh at my friends. So there's myself and a guy named Josh Brumberg. He's the dean for science. I hired Josh Brumberg. And of course, we started a massive rivalry because what is he? He's a Red Sox fan. So it's a Yankee fan versus a Red. And we have bets go on. He owes me so many lunches from this year because every time you have a series. But I also have in the animal facility a guy named Leonard Ramroop, who is a diehard Met fan. And the Mets a couple of years ago did a trade with the Cleveland Indians. And they got Robinson Cano, the ex-Yankee second baseman. He's doing fine with the Mets and whatever. But they gave up this, a rookie pitcher named James something or other. I can't even remember his last name. But guess what? Who was the guy on the Cleveland Indians who threw a pitch, remember, using the cerebellum to get that pitch to go in, and it's doing this, and it's a changeup, or it's going to hit this part of the plate. These guys have such wonderful control. You basically watch him throw the ball. Urshela hits it 440 feet away. And if you basically watch the replay, and I ask you to watch the replay, you basically look at the long-term effect of after the pitcher threw the pitch, he basically puts his glove into his face like that and does this. And you can basically know, even though you can't hear the word, you basically hear the word fuck, and that's it. That he didn't get the ball where he wanted it to go, and Urshela hits a grand slam home run. And a lot of that we're dealing with in that arcane thing of baseball is a lot of cerebellar control here in terms of how you release, where you put your arm, what you're doing. And for those of you who love tennis and watching tennis, or those of you who love to watch golf, I hate golf. It's a waste of space and everything. The point is, what you're having is not only incredible motor control, but incredibly control by the cerebellum. And here we are at 12:04. We're done. So what we're basically going to do, remember, a lot of what we're covering today is not on the first exam. It's only through the corticospinal tract. And what you're going to do within the next little bit of time is talk among yourselves to tell me when I should be sending out part 2. For those of you who have not made an appointment with me on part 1, I obviously have a lot of a slot still open. Send it to me. I answered everybody and answered a few people today. And I'm hoping that this whole process is going to be done by the 23rd of October in this extremely remote environment that we're doing. And then anyone who has any kinds of questions before you get the exam, I'm not going to answer questions during the exam period because that would be unfair, but I'll answer things before that you want to know this or you want to know that, and write me emails. Okay? So have a good day, and I'll see you next week and expect to hear from all of you. Bye-bye.
Thank you.
Thank you, John. I hope just tell me if you thought that the recording was successful, okay?
I will. I'll share the link.
Thank you very much.
Thank you.
Have a good day. I'm ending the session.